Figure 2 illustrates how external hazards and other aspects of the life histories of different animals has impacted programs for tissue maintenance, which consequently influence carcinogenesis. In Part A, the green curve indicates the percentage of the animal population surviving at different ages. The green arrows represent the impact of predation and other external hazards on animal survival. I'll note that predation need not be the primary threat to survival. For example, in wild mice, death by exposure to the cold or through starvation is more common. Given the low numbers of animals surviving to older ages, natural selection has acted to maintain body fitness through periods of likely reproductive success, but not beyond these periods, leading to declining body fitness at older ages. Body fitness is illustrated with the blue curve, and the force of natural selection to maintain fitness is noted with the blue arrows. Note that natural selection does not actively promote fitness decline, but it is the absence of selection for prolonged maintenance that leads to this decline. We can now consider what would happen if the environment changed. As shown in Part B, a change of the same population of animals to an environment with less predation or other external hazards will lead to selection for animals with different life histories. Since a significant number of animals are now living longer and have the potential to reproduce at older ages, natural selection has acted to extend the maintenance of body fitness to older ages, denoted with the open blue arrows. Keep in mind that this evolution of delayed senescence for a mammal would likely take thousands of years. Investment in prolonged tissue maintenance would pay off in terms of reproductive success at later ages. Basically, natural selection tunes somatic maintenance to optimize reproductive success. When we study diseases like cancer in modern humans living in industrialized nations, or when we study mice in our labs, we need to remember that these conditions diverge substantially from those under which both humans and mice evolved. For example, while mice can be maintained for several years in captivity, survival past 6 months in the wild is rare. The curve for maximum lifespan in captivity or for modern humans likely represents a proxy for fitness decline, as survival is now largely but certainly not entirely limited by the evolved programs of tissue maintenance. As we have argued throughout this paper, the decline in tissue maintenance in old age will promote selection for oncogenic events adaptive to the new environment. The expansion of cell clones with adaptive oncogenic mutations will then increase the odds of further progression to cancer. Thus, we can understand how evolved strategies for somatic maintenance to maximize reproductive success lead to increased cancer incidence in old age, explaining why cancer incidence scales with potential lifespan for different animals.